Assalamualaikum warahmatullahi wabarakatuh. My name is BINTI WAHID. So today I want to present about stem and root disease of rubber. I want to talk about stem disease, which are bark necrosis and pink disease, while for root disease I will present about white root disease and purple root disease. So for bark necrosis, the necrosis of the rubber tree is a stem affliction. It manifests itself as a degeneration of cortical tissues at the level of conducing latex tissues. The cola begins right at the grafting point, which is in the transition zone between the trunk and the roots. It causes latex production to stop as well as variation in latex biochemical properties and affects the tree vascular tissues. The root development of necrosis affecting rubber trees is less than ideal. The causal agent for bark necrosis has not found, but showed that bark necrosis is favored by a combination of various stresses. It results from the accumulation of climates and human-induced stresses and physiological malfunction in the grafted trees. Early bark necrosis symptoms develop in the inner phloem tissues but never affect the cambium. Necrotic patches appears in the outer phloem, inducing bark cracking and peeling, but this alteration never lead to tree death. Bark necrosis spread gradually to neighboring rubber trees, and evidence of linear disease centers suggests that a pathogen may be involved, possibly transmitted by the tapping knife. The control management for this bark necrosis is by using chemical and biological control. Next is pink disease. The causative agent for pink disease is corticium salmon color, which causes the death of branches. This pink disease is a disease that affects the stems and branches. The disease is named salmon colored because of its scattered or coalescence pustules and corticoid basidiocarps that are formed on the branches or stem of trees in advanced infections. Usually, it is most serious disease in the young rubber trees. First, at initial stages of infection, it will appear as drops of latex and silky white mycelial growth on the bark surface, and later it will appear pink color. So next, there will be more damaging for plant in the age group of 2 to 12 years. Next, main seat of infection is usually the fork region. The last one is rooting, drying up, and cracking of the affected bark. Then, sprout develop from below the affected portion. Control management: first, use prophylactic treatment. Second, use cultural control, which are clean the preparation or sanitation and maintaining a clean nursery or plantation, do a proper plant density, and others. The third one is mechanical control, which pruning. Next, physical control with proper spacing of seedling for sunshine and aeration, smudging, light trap for pathogen that carrying insects. Then, the use of biocontrol agent and biopesticide, which are antagonists and botanical plant, which also include the effective microorganism (EM). And the last one is by using the chemical control by spraying and swabbing or drenching the fungicides. For white root disease, the causal agent is Rigidoporus lignosus. This fungal organism was first reported on rubber in 1904 from Botanical Garden, Singapore. Its order is Basidiomycete and its family is Polyporaceae. The symptoms for this disease is first, root infection, colonization within the taproot and other part of the root. Second, the foliage discoloration. And the third one is tree branches die back. And the fourth one, the tree is eventually dies. Next, purple root disease. The occurrence of purple root disease is caused by Helicobacterium compactum. Is reported for the first time on Heve basiliensis in India. The infection on nursery plants occasionally leading to their mortality. The symptom for this purple root disease is first purple colored fungal growth on the root surface. Second, adventitious root formation. The third one is the presence of distinct fruiting bodies girdling the collar region. For control management of root disease, first, for immature rubber, use chemical control. For mature rubber, use isolation of branch. For completely killed and dried root, we must prune off and burn it. And for dried plants, we should be uprooted and destroyed. Apply fungicide by painting or drenching, and we can also apply cultural control by replanting the plant using full mechanical clearing method to clear all trees from the land, determine the residual level of inoculum.